So if we have a negative feedback, we also have a positive feedback. So a very good example is the mom giving birth to a child. So first is there's contraction of the uterus. So a woman in labor, of course, the uterus should contract. So once the uterus contracts, the cervix also opens. So it will open. And the stretching of the cervix is the stimulus that will cause the receptor to be activated. And this receptor, because of the stretch of the cervix, it will send signal, an input to the brain, to the control center again. And the brain will now send an output by releasing a hormone. Example here is the oxytocin. The oxytocin will influence the effector organ. The effector organ here is the uterus. So the uterus will contract further. Remember, the stimulus we had is increasing contraction of the uterus. So the uterus will further contract. It will further amplify the contraction of the uterus. So that's a positive feedback. So the first stimulus is uterine contraction. The response is further uterine contraction or further stretching of the cervix, further opening of the cervix. So those are both same stimulus, same endpoint. So that's why we call it positive feedback mechanism. Clear? Okay, we can actually move on again. So we've talked about negative, positive feedback based on response. And based on level of involvement, you have your local control system, systemic control system. When you say local, it's occur in the area of the stimulus. Systemic involve regions distant from the area of the stimulus. So this is your systemic control system. So this is an example. Again, I've been constantly emphasizing the different components of your control system. That guy, I'm not that one. I look better. Local versus reflex control. So local is you have a local change, a local response, and a local change in that particular area. But for systemic, for reflex control or systemic, it goes all the way down from the receptor to the brain and down to the effector. So it's a long route compared to the local. So the organism in homeostasis, you have external, internal change, and any of this can result to loss of homeostasis. And we try to bring it back or to compensate by those external changes. But if we fail, it will result to illness or disease. But if we succeed, it will lead to wellness. So this slide is important in a way because the cell can do all these things because of the ability of the cell to have cell-to-cell communication. And this is through intercellular and chemical messengers. So systemic control system, the hormones are secreted by a cell and uses blood as a delivery system. Again, these hormones, neurotransmitters, are actually the carrier of the input or the output. So they are very important because if you don't have those, they will not be relayed to the brain or they will not be relayed to the effectors. So hormone secreting gland, it will secrete your hormone, will cause an effect on the blood vessel or your target cell. Your nerve cell can also secrete neurotransmitter and cause an effector organ to elicit response, and can also be releasing hormone coming from the nerve cell affecting a particular blood vessel or a target cell. Okay, so these are your chemical messengers. Take a look at this. It's local messenger secreted by a cell which binds to a nearby cell. Autocrine agent, messenger secreted by a cell which binds to its own plasma membrane, auto, self. So these are examples of your local control system. So they are also example of chemical messengers. So again, local or systemic. This is an example of a local control system. So going back to the question, what will be your answer? Yes, correct. I will guess, I think there will be Michelle in the class. So Michelle, you are correct. Whoever you are, I hope to meet you soon. So the answer is letter D, systemic negative feedback. Okay? Systemic because it goes a long way, travels to the brain and to the kidneys. And negative because of opposite direction of your stimulus and your response. Okay, another question. Which of the following scenarios will cause a stable water balance? So you have here A, B, C, and D, and water intake in liters, water from metabolism also in liters, and urine also in liters. So for letter A, you have 2.5, 0.5, and 2.0. Letter B, 2.0, 0.5 liters, 2.0. Letter C, 2.5, 0.5, 3.0. Letter D, 2.0, 0.5, and 3.0. So the question again is, which of the following scenarios will cause a stable water balance? So storage pool in the body is affected by two events. One is net gain, and the other one is net loss from the body. So from the net gain, you have food coming from the GI tract, air from the lungs, and synthesis in the body as well, or those being metabolized. Net loss, you have from metabolism and also from excretion from the lungs, GI tract, kidney, skin, as well as your menstrual flow. Okay, so the homeostatic systems regulate balance between addition and removal of substances. So these are your net gain and net loss from the body. So just to give you a more concrete example of water gain and water loss, so from food and drink, there you can see almost more than a liter per day. And also there's metabolism that contributes to the gain of water in the body. And we have your intake as well. For the loss, usually you have your insensible and sensible losses from the skin, lungs, urine, feces as well. So this will greatly affect the amount of water in your body. So this has to be balanced, gain and loss. So bringing back the slide, in your body, the amount of water you have is basically 42 liters. Okay, 42 liters. And then for your net gain or water input, you have coming from fluid and food, from synthesis, a total amount of 2,500 liters. And for the net loss, insensible, sweat, feces, urine, a total of 2,500 liters or ml as well. So what do you observe from this slide? Whatever you take, any excess will also be excreted, meaning you just have to maintain the 42 liters needed by your body. So this is the amount of water you need. Anything in excess, it will be excreted. So that is now your normal water balance. So for example, your gain is equal to the loss. You have a stable water balance or stable balance. And you have a normal body store. Okay? Because it's equal. You have gain the same as your loss. But what if your gain is higher compared to your loss? That is now what you call positive balance. And what happened to your body store? It goes up. Okay? We go to the opposite. If your loss or your output is higher, your balance now is termed as negative balance. What happened to your body store? It goes down. Clear? Okay. So this is gain, loss, balance, and what happened eventually to your body store, you have your positive and negative balance. So if we go back to the question, what do you think will be the answer? Michael? May Michael ba dito? Okay. Very good, Michael. The answer is letter C. So 2.5 plus 0.5 is 3. So that will be your gain. And then your urine, you excreted 3 liters. So this is now your stable water balance. So we go to the body fluid compartments. So the total body water is computed by multiplying your body weight by 0.6. Or to put it in a different term, usually the total body water comprise about 60% of the body weight. And they are distributed intracellularly and extracellularly. Intracellular fluid, extracellular fluid. 40% of the body weight is intracellular. 20% of the body weight is extracellular fluid. Extracellular fluid is further divided into interstitial and your plasma. So when you talk about interstitial fluid and plasma, they belong to extracellular fluid. Okay? And the 20% of the extracellular fluid is distributed in the interstitial, about 0.75 or 2/3 of the ECF, and 0.25 of the ECF is plasma. Okay? So put numbers on it. So let's assume you have a 70 kilogram man. 60% of the body weight of that individual will be 42 liters. The ECF, we say it's 20%, so that will be 14 liters. And intercellular will be 40%, so that will be 28 liters. How about the ECF? We have the plasma and the interstitial fluid. We say it's 3/4, 1/4, or 10.5 liters for the IF. Plasma is 3.5 liters. So this is now how your body fluids are distributed in the body. So you have your ECF, ICF, your ECF, you have your IF, and your plasma. The plasma, we're talking basically what's in the blood. Okay? So cells regulate their activities by maintaining the composition differences across the cell membrane. So there are barriers actually between the ECF and the ICF. So if you look at the ECF and the ICF, there is the cell membrane. We've talked a lot about cell membrane a while ago. And between the IF and the plasma, you have another membrane, a specialized type of membrane. It's a very thin wall. It's called your capillary wall. So there's an exchange of fluid between the two compartments. So that membrane separates the membrane of the ICF and the ECF. The plasma and the interstitial fluid separated by the capillary wall. Important also for you to understand or remember are the components of the different solutes in the extracellular and intracellular component. For example, in extracellular, sodium is the most abundant, cation, positively charged, so it's 145, less amount in the intracellular. If we go to potassium, it has more concentration on the intracellular, meaning more potassium are inside the cell, which is 150. So the major extracellular cation in the extracellular is sodium. The major intracellular cation of the cell is your potassium. So these are the major solutes or cations in the extra and intracellular cell. And we have the rest of your chloride, which is more on the extracellular as well, and the different solutes located in the extracellular, intracellular components. So another type of question in physiology is what you call as quantitative comparison. So write A if the item is greater than item 2, B if item 1 is less than item 2, and C if items 1 and 2 are equal. Example, we have a sodium. 1 intracellular, 2 extracellular. So which is greater? Is it in the intra, extra? How about potassium? Intra, extracellular as well. So I think this is again a hanging question for you guys. So I'm giving you more chance to win a prize at the end of the lecture. So this is QC or quantitative comparison. Down to my last topic is your mechanisms of membrane transport. Question, the movement of sodium in a sodium-potassium ATPase pump is along both its concentration and electrical gradient, against both its concentration and electrical gradient, along its concentration but against its electrical gradient, against its concentration gradient but along its electrical gradient. We're going to answer that later on. So membrane transport proteins, water channels, ion channels, can be ligand-gated, voltage-gated, mechanically gated. Solute carriers, you have uniporters, symporters, antiporters, ATP-dependent transporters. Water channels, if we talk about water channels, they are the main route of water movement into and out of the cell. And you will always encounter this channel called aquaporins. When we're talking about water movement out to inside of the cell or inside to outside of the cell, we were going to encounter aquaporins. So when you talk about water channels, it's aquaporins. So it can be regulated by altering the number of aquaporins in the membrane or by changing their permeability. So this is water channels. We are now talking about membrane transport proteins. Okay? So that channel is an example of a membrane protein. So how about ion channels? We say they can be ligand-gated, voltage-gated, mechanically gated. Important for the function of excitable cells like your neurons and muscle cells. This will be your next topic. Types are based on selectivity or non-selectivity. Example of selectivity, it can only be for one specific solute. The other one will be, it can allow passage of different solutes. It can also be based on conductance, meaning higher concentration gradient, and gating. So when you say ligand-gated, they open when a specific molecule binds to it. And then voltage-gated, open when there is a change in membrane potential. So it has something to do with the membrane potential. Mechanically gated, when there is physical deformation of the membrane. So the channel will open up. So how about solute carriers? Can be uniporters, symporters, antiporters, and ATP-dependent transporters. So when you say uniporters, they transport a single molecule across the membrane. So one, single. So an example of this is your GLUT2 transporter. It transports glucose into the cell. So in this picture depicted, you allow the entry of glucose, only one molecule, and that transporter there, that membrane protein, allows only glucose to move from one compartment to the another compartment. How about symporters or co-transport? They are both the same. So it couples the movement of two or more molecules, ions, across the membrane. Take note of this, they are transported in the same direction. So same direction. An example given here is your sodium glucose transporter. So the cytoplasmic space, and you have your ECF. So if you look at the movement, the sodium and glucose moves from the extracellular, meaning from the plasma, goes inside the cell by this symporter. The name of the symporter is your sodium glucose symporter. They are symporter because they move in the same direction. Antiporters, exchanges, counter-transporters are the same. It's basically the opposite of symporters. For example, is you have your sodium chloride, sodium calcium exchanger, and your sodium hydrogen exchanger. It couples the movement of two or more molecule ions. However, one is transported in the opposite direction. Okay? Sodium is going in, and the chloride, the calcium rather, is going out. So, like in the sodium hydrogen, the sodium is going in, hydrogen is going out. So it's in the opposite direction. ATP dependent, from the word itself, it uses energy, and the energy here is your ATP. It drives the movement of molecules, ions across the membrane. So in this example, you can see that sodium moving out from the cell, and then potassium going inside the cell. So the important thing here to remember is that it utilizes energy in the form of ATP. So we mentioned channels and carriers, and these are transmembrane proteins that helps facilitate the movement of solutes from in to outside of the cell or outside to inside of the cell. So what are the differences between channels and carriers? One is channels are intermittently open. Carriers, they are never open. Unitary event, channels just open up and then transport the solute. But in carriers, remember this, it has to undergo conformational change. I will give you an example later on, but just take note of this. Carriers undergoes conformational change. Particles translocated, for channels smaller, carriers usually it's bigger. The translocation rate for channels, it's rapid, carriers is slower. Saturability, channels, there's no saturation. It can transport as many as possible. But for carriers, you have to remember, it has a saturation point. It can be saturated. Okay? So please take note of this slide, a very important slide as we discuss more about membrane transport proteins in the next couple of minutes. So, how does this cell move from one compartment to another? So we talk about the concept of random thermal motion. So in random thermal motion is synonymous with Brownian movement. So meaning molecules are always moving. So when molecules are always moving, they collide. So when they collide, they bounce off, they bounce from each other. So bouncing of these molecules, they will collide, and this collision will lead now to generate energy. And this energy pushes molecules from one direction or another. So this is an important concept wherein the movement will predict the molecules. So from high energy state to low energy state. So there should be a driving force for these molecules to move. This can either be chemical, electrical, and electrochemical. So we're going to talk first about the chemical. So a question, is there a concentration gradient illustrated in figure A? The red dots illustrates the solutes. The blue background illustrates the extracellular fluid. So the blue is your ECF. The tan color within the cell is your ICF. The red dots again are the solutes. So the question, is there a concentration gradient illustrated? The answer should be yes, because there is more solutes inside the cell. So this higher number of solutes inside the intracellular fluid compared to a lower number of solutes in the extracellular fluid. So a high frequency of collusion inside the cell will create more energy and forcibly push solutes out of the cell. So the movement now will be from higher concentration to lower concentration. How about in this figure B? Again, the same illustration, but there's a higher concentration of solutes the outside of the cell, meaning lower concentration inside the cell. So since more energy is being produced at the ECF, so we will now predict the movement. It will be from higher concentration to lower concentration. So from the ECF to the ICF. So this is the chemical driving force. To emphasize again, it should be from higher concentration to lesser concentration. Now we talk about electrical driving force. The electrical driving force deals with solutes that have charges. So you have your negative and your positive. In the ICF, there is negative charge, meaning you have more negatives inside the cell. There are more negative charged ions in the ICF. For the ECF, there is net positive charge. But throughout this topic or lecture, we will depict the cell something like this. So your ICF has a negative net charge. So the inside of the cell will be more negative. And the ECF will have more positive net charge. Okay? So this is the ECF and the ICF with the negative and positive charges. Is there an electrical gradient? The answer is yes. There is a difference in charges across the membrane, or there is a charge separation across the membrane. So look at this figure. We focus on the letter A. So electrical driving force, if we have a net positive charge in this ECF and a net negative in the ICF, why do the positives want to move in? The positives are attracted to the negative charge in the ICF. Remember, opposite attracts. So in letter B, the negatives are attracted to the positive charge in the ECF. Again, opposite attracts. So electrical gradient and not diffusion. So positive going inside the cell, negative going outside the cell. That is electrical gradient. So we've talked about concentration gradient and electrical gradient. So which has a higher concentration of sodium? Is it ECF or ICF? Yes, sodium has greater concentration on the ECF. So the movement should be from higher concentration to lower concentration. So your answer should be sodium. Okay? Sodium going inside the cell based on chemical driving force. So membrane transport, you have different types. When you say movement is along a gradient, it is considered passive. 3 types: simple diffusion, facilitated diffusion, osmosis. Movement is against a gradient. It's active. There are 2 types: primary active and secondary active. Movement requires a vesicular, 2 types: endocytosis, exocytosis. And movement requires a vector, epithelial, since it is seen in epithelial cells, 2 types: paracellular and transcellular. So these are your membrane transport: passive, active, vesicular, and epithelial. And I've shown you the different examples of each different membrane transport systems. We go first to the mediated transport system. uses an integral protein known as transporter or carrier in transporting a molecule across a membrane. Transportation across a membrane requires three processes. Take note of this, it's very important. Binding of the transported solute to a carrier, conformational change of the carrier, release of the transported solute to the other side of the membrane. So these three steps should be met in order for a solute to move from inside to outside or outside to the cell in mediated transport systems. An example of mediated transport systems are your facilitated diffusion and active transport. Okay? Facilitated diffusion and active transport. First is diffusion. Diffusion is a process by which molecules move spontaneously from an area of high concentration to low concentration. So just remember, DHL. I'm sure you are familiar with DHL. Diffusion, high concentration to low concentration. The movement is actually secondary to the random thermal motion. We've discussed that a while ago. And the warmer the environment, the more rapid is the movement. Okay, that's diffusion. And there are factors that will affect diffusion and then can be explained by the different equations on your screen. You have fixed first law of diffusion, Stokes-Einstein equation, Van der Waals equation. And the first law, or the fixed law, net diffusion, you have the numerator and the denominator. Diffusion coefficient, the area, concentration gradient over the thickness. And how you interpret this one is, on the numerator, if you increase the diffusion coefficient, the area, the concentration gradient, your diffusion will be faster. Okay? But if you increase the thickness, diffusion will be slower. So that's how you interpret or analyze the fixed law and how it affects your diffusion. And what are the factors that will affect your diffusion coefficient? So this is depicted in this equation, your Stokes-Einstein, Boltzmann constant, temperature, the radius of the molecule, viscosity of the medium. So on the numerator again, if you increase the temp, the constant, you increase diffusion coefficient. If you increase diffusion coefficient, you increase diffusion. Okay? How about radius and viscosity? This will greatly affect your diffusion coefficient and eventually slowing down your diffusion. Okay? Clear so far? So in Van der Waals equation, you also have your net flux, which affects also your diffusion, permeability constant, membrane surface area, and your concentration gradient. So these three factors will increase your net flux. If you increase your net flux, you also increase your diffusion. So another type of question in physiology is your variation relationship. Write A, if an increase in item 1 is accompanied by increase in item 2. Write B, if an increase in item 1 is accompanied by decrease in item 2. C, if an increase in item 1 is not accompanied by any change in item 2. I provided you again the diffusion rate equation here. Number 1, concentration gradient and net diffusion. What do you think is the answer? Number 2, thickness of the membrane and net diffusion. So if we increase item 1, what will happen to the net diffusion? The answer is A, increasing the concentration gradient, increase the net diffusion. What will happen to number 2? Thickness of the membrane and net diffusion. The answer is letter B, because if we increase the thickness, the diffusion will decrease. Okay? And lastly, net diffusion coefficient, what will happen? I hope you all answer letter C, because it should be the other way around. Diffusion coefficient should cause an increase in the net diffusion, not net diffusion causing increase or decrease in the diffusion coefficient, because the net diffusion is the one that is affected by your diffusion coefficient. Okay? Meaning they are unrelated. Look at the letter C, explanation for your variation relationship. Okay, we move on. Simple diffusion is a kinetic movement occurred through a membrane protein or two intermolecular spaces without any interaction with carrier proteins in the membrane. Increasing the concentration of diffusing substance will increase diffusion. Example, aquaporins, but aquaporins, I want you to remember at this point that it is a water channel, and we're going to talk more about this when we talk about osmosis. So we're going to talk about simple diffusion. So you have your phospholipid bilayer, the plasma membrane. We have a solute that can possibly pass through the membrane, and we label this as X. And this solute, or your X here, can freely pass through the membrane. It means it has high permeability. So the core or the tails contains fatty acids. So these fatty acid tails are fats. Anything that is fat soluble will freely diffuse through the membrane. If it is fat soluble, they are lipophilic. Philic means to love, lipo means fat. So fat soluble substances are lipophilic or fat lovers. If fat soluble substances love fat, they will be afraid of water. So they are hydrophobic. Hydro means water, phobic means fear. Let's talk about water here. You have your H, hydrogen ions, and your O, your oxygen. Okay, so your H has charges, your O has also charge. So any molecule that is nonpolar will have no partial charges. So if there is no partial charge, it is not attracted to water. So they are non-polar molecule and they are hydrophobic. So substances that can freely diffuse include: one is lipophilic, second, hydrophobic, third, you have your non-polar solutes. What about small polar and large polar? Meaning they have charges. So small polar molecules can diffuse due to their size. An example of which is water. Even though they have partial charges, they can still pass through because of the small size. So small polar molecules like your water can pass through the membrane. But here's the catch. In this course, if you are talking about the movement of water, aquaporin must be present, and we are talking about osmosis. So throughout your course, when we talk about osmosis, we are talking about movement of water. We're not talking about diffusion. We're talking about osmosis, movement of water through aquaporin channels. Okay? So osmosis is not simple diffusion. Clear? Then we can move on. So one example of passive transport is your facilitated diffusion. We've talked about simple diffusion. Now it's facilitated. Why facilitated? It requires interaction with a carrier protein. Remember this. Facilitated requires carrier protein. And the net flux of molecules across a membrane always proceeds from higher to lower concentration. The rate of diffusion approaches a maximum Vmax. So it is a Vmax. Example, transport of glucose and amino acids. Emphasis again, higher to lower concentration, and a carrier protein is needed for facilitated diffusion. This is again an illustration on how facilitated diffusion is, which we label it. This is your ECF, your ICF, extracellular fluid, intracellular fluid, and you have your plasma membrane or the phospholipid bilayer. And an important structure is the transmembrane protein. This is your carrier protein. So step one, first step. First, we have the solutes scattered at the ECF and the ICF. What do you notice? Higher concentration at the ECF and lower concentration at the ICF. So how will we predict the movement? Again, we say diffusion from higher concentration to lower concentration. So the high concentration in the ECF will cause collisions and generate energy. This energy will drive that molecule and pass through that membrane. But the molecule is too large to freely diffuse. So the molecule maybe is polar. So we need a carrier protein. Okay, so that carrier protein will now accept the solutes or binds to the carrier protein, and look at the carrier protein. It fits perfectly with the solute. So what does it mean? It has a specificity. So it's very specific for that solute. Only certain carrier proteins can transport certain solutes. Okay? So you need a carrier protein that's very specific. In this example, it's specific for glucose. Okay? It's very specific for glucose. This is your GLUT transporter. So going back to the figure, your solute now binds to the carrier protein. So what will be the next step? The next step now will be there is conformational change. Look what happened to the carrier protein. It transformed into different shape. Again, illustrates specificity. That conformational change now will deliver that solute into the ICF. And then once solute is now in the ICF, the next step will be reverting that carrier protein to the original shape and again ready to accept another set of solutes. This example illustrates how solutes are brought into a cell, and this carrier protein can also be used to move solutes out of the cell. Carrier proteins will assist large polar hydrophilic or lipophobic solutes. So that's why you need this carrier protein. Another concept here is it undergoes conformational change of the carrier protein to deliver the solutes into the cell or outside of the cell. So this is now an example of facilitated diffusion. How about diffusion through channels? So ECF again, ICF being labeled for you. So for example, you have a solute that goes through the channel. Again, that's very specific for one channel. So channel proteins also exhibit specificity. For example, you may have a sodium channel. Only sodium will pass through the channel. It's a sodium channel, very selective, very specific. Or a potassium channel, so it passes through the membrane. So channels also exhibit a specificity, much like carrier proteins, but there is no conformational change that happens as opposed to facilitated diffusion. There are also different types, okay? Some channels are always open, like your leak channels. We'll talk more about in your next lectures about leak channels. They're open most of the time. And some channels are conditionally open, or we call it gated channels. So gated, open or closed. There are different types of gated channels. Again, we're going to talk more about this in your next few lectures. So another example of passive transport is osmosis, the movement of water across a semi-permeable membrane. Water moves from a compartment of greater water concentration to a compartment of lesser water concentration. Again, from greater water to lesser water concentration. Or from a low solute concentration to a high solute concentration. Because in low solute concentration, it has more water. High solute concentration, it has less water. So that is the process of osmosis. So which direction will the water move? assuming we have a non-permeating solute. A concept here is non-permeating, meaning a solute will not be able to cross the membrane. So non-permeating. Water moves from high to low water concentration. Basically, that's why we have to remember about osmosis. So you have two solutions here. Side 1, side 2. The red one or the solute, solvent, or the blue one, or the water. So what will be the movement in osmosis? Again, remember, higher to lower concentration. So the movement will be from side 1 to side 2. So what do you notice? There is an increase in the volume on the other chamber, or inside side 2, because water moves from side 1 to side 2. So remember the osmosis, volumes change when water diffuses. So that is the process of osmosis. We've discussed osmosis. We're going to talk about also osmolarity. So osmolarity is defined as the total number of solute particles, osmoles per liter of solution, and water concentration is indirectly measured through osmolarity. So this is the unit of when we talk about osmosis. So osmolarity, total concentration of solute per liter of solution. So larity is liter of solution. Osmolality is total concentration of solute per kilogram of solvent. So what do we use as a unit? We use osmolality because this is the one that is not affected by temperature. So osmolality. So in your next lectures, we're going to use more osmolality rather than osmolarity. So we've talked about osmosis. So how will that water move from high concentration to low concentration? It has to have a pressure. So that pressure is termed as your osmotic pressure. Okay? Osmotic pressure is depicted here by your Van Hoff's law. So to have the equation here, we have the osmotic pressure. Reflection coefficient is basically telling us the permeability coefficient, the number of dissociable particles per molecule, total solute concentration, the gas constant, and the temperature in degrees Kelvin. So this is your Van Hoff's law. So when you encounter Van Hoff's law, we're talking about osmotic pressure. The osmotic pressure is the pressure that drives water to move from one compartment to the another compartment. Okay? So that is your osmotic pressure. Osmolarity, we defined osmolarity a while ago. So it also depends on the number of particles in the solution. So we have two solutions here. The first one contains glucose, only one solute, and the other one is your sodium chloride. So for glucose, it only contains 1 osmol per liter, and for sodium chloride, it has 2 particles or 2 dissociable particles. So you'll have 2 osmoles per liter of solution. Okay? So that is what you mean as it depends on the number of particles in the solution. So I mentioned a while ago about permeable and non-permeable. Permeable is also synonymous with penetrating, and non-permeable is synonymous with non-penetrating. So we've mentioned permeability of the solutes, okay, or reflection coefficient is a dimensionless number of this permeability. So if we label it as 1, it means it's completely impermeable, 0 completely permeable. What does it mean? When you say impermeable, it is non-penetrating or non-permeable, meaning the solute cannot cross the membrane. It does not penetrate the membrane. And if it's 0, the solute can penetrate the membrane. So when you say permeable, it's penetrating or it's very diffusible. Because we need to create volume change in one solution. We're going to talk more about this, the tonicity. When we talk about tonicity, is the ability of that solution to change the volume of the cell. Okay, we're going to talk more about that later on, but at this point, you know that the concept of permeable and non-permeable solutes. Example, in osmosis and permeable solutes, so you have initial state of a solution and equilibrium state. In the initial compartment, the osmolarity is different and the equilibrium is the same. Okay, we dissect the initial solution, the purple ones, the solute. In the solution 1, you have 2 osmol. Solution 2, you have 4. Water content, 53.5, 51.5 for compartment number 2. But the volume is the same. Because we say, when you have more solutes, what do we say? We have less water. When you have less solute, we have more water. But we're talking about here a permeable solute that can penetrate the membrane. So the solute now, the solutes will penetrate the membrane. What does it do? It tries to equilibrate. It tries to equilibrate. Okay? So after equilibrium, what will happen now to the compartment's osmolarity and volume? It will now eventually be the same. From the initial state of having different compartment osmolarity, but the same volume, when the solute moves to the other compartment because it was able to penetrate, or it is a permeable solute, at the equilibrium state, it has now the same osmolarity. Okay? So the concept here is when you have a permeable solute, it will just try to balance. It will not create a change in the volume of that particular compartment. So what does non-penetrating solute do with the osmolarity and the ECF and the ICF? So when you talk about non-penetrating, non-permeable, these are the solutes that will not be able to cross the membrane. Example of which is your sodium chloride, potassium, and your organic acids. Remember that the osmolarity between the ECF and the ICF are equal, meaning if they are equal, there will be no movement of water from ICF to ECF or ECF to ICF. So you maintain that normal plasma or that normal osmolarity in each compartment, non-penetrating solute. So we have a term called isoosmotic, hyperosmotic, hypoosmotic. So when we talk about osmotic, we're talking about penetrating and non-penetrating. Isotonic, hypertonic, hypotonic, non-penetrating solution only. Because when you talk about tonicity again, it's the ability of that solution to change the volume of the cell. When you talk about osmotic or the permeable, it does not have the ability to change the volume of the cell. Okay?